To download MGL Tools, search for MGL Tools on Google and open the official MGL Tools website. Click on the download option. The software is available for Linux, macOS, and Windows operating systems. Select your operating system and click on the corresponding download link to download the software package. MGL Tools is used for preparation of protein and ligand structures, grid generation, and performing molecular docking using AutoDock. After downloading MGL Tools, we will download AutoDock 4. AutoDock 4 is used to predict the binding interactions and binding affinity between protein targets and ligand molecules through molecular docking studies. Search for AutoDock 4 on Google and open the official AutoDock webpage. Click on the download link and select the appropriate version for your operating system to download the software. AutoDock 4 is integrated with MGL Tools, which provides the graphical interface and utilities required to perform molecular docking studies. Let the MGL Tools and AutoDock 4 download in the background. Meanwhile, we will proceed to download UCSF Chimera. Search on Google for download UCSF Chimera and open the official UCSF Chimera webpage. The software is available for multiple platforms, including Windows, Linux, and macOS. Download the appropriate version according to your operating system. UCSF Chimera is used for multiple purposes, including molecular visualization, target protein preparation, structure analysis, and energy minimization for molecular docking studies. Now we will download LigPlus. Search on Google for MGL LigPlus and open the official LigPlus webpage. Go to the academic license section and fill in the required details such as your name, institutional address, and academic email ID. Use an institutional or academic email address for registration. Personal email services such as Gmail, Yahoo, or Rediffmail may not be accepted for academic license access. The user ID, password, and download link for LigPlus will be sent to your registered academic email ID. Open your email inbox and click on the download link provided in the email. Enter the registered email ID and password to download LigPlus. LigPlus is used for generating 2D ligand-protein interaction diagrams, including hydrogen bonds and hydrophobic interactions between the ligand and target protein. Now we will install these software packages on our PC. First, we will begin with the installation of LigPlus. LigPlus is downloaded in a compressed zip format. After downloading, right-click on the zip file and select Extract Here or Extract to LigPlus using extraction software such as WinRAR or 7-zip. Once the extraction process is completed, a new LigPlus folder will be created. Copy the extracted LigPlus folder and paste it directly into the C drive of your computer for easy access and proper software functioning. Ensure that Java is installed on your PC before running LigPlus, as the software requires the Java Runtime Environment, JRE, for execution. Double-click on the LigPlus icon to launch the software. A setup window may appear asking for path and directory configuration information. Review the settings and click OK to continue with the initialization process. Here, you need to create a folder named Temp in the LigPlus folder. During the initial setup of LigPlus, set the LigPlus temporary directory path to this folder. This temporary directory is required for proper execution and file processing in LigPlus. After clicking on Save, LigPlus is ready to use. From the File menu, you can open any protein-ligand complex structure in PDB or DRW file format for visualization and generation of 2D ligand-protein interaction diagrams. Now we will install MGL Tools on our system. Open the downloaded MGL Tools setup file to begin the installation process. A setup wizard will appear on the screen. Click on Next to continue. Accept the license agreement by clicking on I agree. Select the installation directory or keep the default installation path recommended by the setup wizard. Continue clicking Next to proceed with the installation. Wait until all required files are installed successfully. After completion, click on Finish to complete the installation process. MGL Tools, including AutoDock Tools, ADT, is now ready to use for molecular docking studies. The current version of MGL Tools alone is not sufficient for performing molecular docking studies, so we will install AutoDock 4 for proper integration and docking execution within the MGL Tools environment. Now we will install AutoDock 4 on our system. Open the downloaded AutoDock setup file. A setup wizard will appear. Click on Install to continue. After the installation is completed successfully, click on Close to exit the setup window. Now we will install UCSF Chimera on our system. Open the downloaded UCSF Chimera setup file. Click on Next to continue. Check the options, create a desktop icon and create a quick launch icon, then continue clicking Next to proceed with the installation process. Wait until the installation process is completed successfully. Once the progress bar reaches completion, click on Finish to close the setup wizard. After installation, UCSF Chimera is ready to use for molecular visualization, protein preparation, structure analysis, and energy minimization in molecular docking studies.